Hi, I'm Dennis Wang. I'm a bioinformatics lecturer at the University of Sheffield, and today I'm going to give you a quick overview about functional analysis, what it is, and why it is important in bioinformatics. So in large genome studies, you often can identify genetic variants in a cohort of individuals that are associated with a phenotype or the disease. So in the case of this Manhattan plot, each dot is a genetic variant, and sometimes you find a few or quite a lot of genetic variants that are associated with the disease. This often requires sequencing a large number of individuals to find variants that are associated with the disease, such as schizophrenia. But identifying associations between genetic variants and the disease is only one aspect of determining whether there are biomarkers for the disease or a particular phenotype. So once you get a genetic variant that has an association with the disease, you still have to collect other evidence to make sure that the variant or the gene that the variant sits in is actually causal or has some function. So therefore, often following genome studies, you conduct a series of functional assays to test whether the genetic variant or the gene has some particular function and may cause the phenotype of disease. So some of those particular assays are transcriptomic assays, epigenetic assays like ChIP-seq, and this talk and exercise will focus more on transcriptional assays such as RNA sequencing. So what are gene signatures? So I'm sure some of you have heard the term of signatures or gene signatures. And basically, the definition of a gene signature is a collection of gene expression profiles that describe a biological process or a condition. So there has been many gene signatures that have been published over the past couple of decades. So in the early days of genomics and expression profiling, um, there has been a lot of studies that basically found genetic variants and genes and their changes in expression that correlate with a disease or phenotype. So for some diseases such as breast cancer, there have been many genes that have been profiled using, say, microarrays or RNA-seq experiments, and they've identified associations between those genes and the disease. So some of those signatures have been clinically actionable and actually became kind of clinical tools now used routinely. So some of the early signatures that have been identified in, say, melanoma and breast cancer have become kind of commercial diagnostic tools, but it's taken a long time. So as you can see from this time scale, it's almost taken a decade for a gene signature to be discovered in a scientific setting and kind of translated into a diagnostic tool is that has kind of healthcare regulatory approval. So there are still many gene signatures that haven't been validated or translated into clinical diagnostic tools. In the instance of colorectal cancers, there has been a number of gene signatures that have been found, but many of them have not been able to be reproduced. So one of the reasons why a lot of gene signatures cannot be validated is because there could be actually quite a lot of different signatures that could actually be identify a particular disease or be associated with a disease. So this is an earlier kind of computational paper that showed that even when you randomly pick groups of genes and their expression profiles, you could still find association with particular diseases. So one of the reasons why you can find random sets of genes that are associated with a phenotype or disease is because genes actually interact with other genes and form higher level structures in terms of modules or networks or pathways, biological processes, and often these have a particular function in a cell or a biological system. So therefore, we should not look at genes at an individual level, but we should look at the interaction between genes or groups of interactions of genes in the form of a network or a pathway, and actually what kind of function that kind of higher level structure has. So you might have often come across this type of question where someone asks you, give me the key pathways that drive this particular disease or phenotype. So one example of why this problem is difficult to answer is when you profile a number of patients, say do RNA sequencing on 4 patients, you might actually identify different sets of genes that are upregulated or downregulated. And you might be able to identify connections between these genes, such as these arrows here, but still unclear how these genes and patients relate to one another. But if we were able to overlay some more functional information about the interactions between the genes, such as the ones highlighted in yellow here are involved in non-like receptor signaling pathways, then we are able to connect some of the patients and genes. So one thing to be aware of is that there are many different types of interactions between genes. Some are interactions between proteins. Some are more statistical interactions like the co-expression of the genes. So if gene A is upregulated in a number of patients and gene B is also upregulated in the same patients, they are considered as being co-expressed. And then there are transcriptional regulatory interactions, for instance, when a particular gene encodes for a protein that binds, say, to a promoter or enhancer region and regulates another gene. So protein-protein interactions are actually kind of physical interactions between genes, and they're often captured in uh, different types of proteomics assays like mass spectrometry that we are able to experimentally observe interactions between proteins. So sometimes interactions between proteins also uh, produce some kind of signal. So for instance, when interacting proteins phosphorylate each other, you could actually get a signaling cascade that may actually activate some downstream processes like apoptosis. And then there are other types of networks or pathways that are metabolic. For instance, interactions between enzymes may actually um, produce some produce certain types of reactions that may generate products or degrade certain products. So some of those pathways are described in a few special databases like Reactome and KEGG, which we will talk a bit more in a second. And an example of gene regulatory interactions is microRNA networks. So microRNAs are RNAs that actually can regulate other genes. And then other types of gene regulatory interactions are those where proteins might bind to DNA and then and regulate genes that are encoded in that DNA. So this information about gene interactions and the formation of pathways are stored in a number of databases like Reactome. Another database that stores information about the functions of gene networks is the gene ontology databases. And this basically groups a number of genes and their interactions into specific functional terms like focal adhesion, ERB signaling, chemokine signaling. Well, one key thing to remember is that also these pathway or network functions don't exist in isolation. So as you can see here, these different functions are connected to each other, and sometimes they overlap with one another, um, or and form some level of hierarchy. So there are many, many more databases that store pathway and network information. Uh, the ones I've listed here are some just the common ones, but you can search for these kind of pathway databases quite easily in any search engine. So to summarize, functional analysis enables you to take information from gene variants, their association with phenotypes and genes, and understand uh, their function to enable clinical interpretation or molecular characterization of the genes that are associated with a particular phenotype. In the following exercise, we'll conduct one type of functional analysis, which is gene set enrichment or pathway enrichment, and we'll use two different tools, Gorilla and GSEA.